I'm actually, if you've never seen me in person, I'm not that tall, so I don't know why our computer makes me look so much taller than Michaela and Shauna.
Big in personality.
But yes, this is the science we're doing here is regenerative medicine to make you taller.
All right.
Woo. So anyway, welcome to our Wednesday morning Facebook Live and other live channels where you may be enjoying our weekly program. It's been a really, really interesting week in neuroscience, and there are definitely things we can chat about. I wanted to spotlight in particular, and this is why I wanted to have Sean and Michaela on with me. They are my clinical trial manager and clinical trial assistants. Some very interesting news that actually came out yesterday. Big press release, hugely anticipated, somewhat disappointing, but a deeper dive leaves us with a little bit of optimism, I think, as to what's going on. Yeah. And that is the results of the Brainstorm Cell Therapeutics Phase 3 Neurone trial, which is a autologous stem cell treatment for Lou Gehrig's disease or amyotrophic lateral sclerosis. This trial was hugely anticipated. Brainstorm Cell Therapeutics basically has one technology, and that's their Neurone technology, in which they take your own stem cells that are extracted from your bone marrow. They use their patented process to manipulate these stem cells to not only expand them so that you have more cells available, but that they express what they call neural elements. So they're basically signaling molecules that stimulate the central nervous system specifically to regenerate, to heal itself. Now, if you go to the Brainstorm Cell Therapeutics website and look at their pipeline, you'll see that there's a lot of interest in applying this technology across the board. This is, again, investigational, not an FDA-approved treatment at this time, but to apply it to conditions as diverse as multiple sclerosis, Parkinson's disease, Alzheimer's disease, and of course Lou Gehrig's disease or ALS. Along those lines, there was an article, it's just a newspaper article that came out that said by the year 2027, the stem cell industry will be a $47 billion industry worldwide. So we know that stem cell technology is not going away. We're certainly very hopeful here about the promise of what stem cell technology can offer folks at not only around the world, but here in Ozark, Missouri at Charlotte Health and Neurology. And we take this science very seriously, so we participate in clinical trials. We have about 6 active trials going on now.
Yep.
So what happened, let's talk a little bit about Brainstorm Cell Therapeutics and Neurone. So first of all, if you're not familiar with how clinical trials work, in general, clinical trials go through different steps where first the compound that's under study, and usually it has sort of a letter and number name. It may not have a fancy drug name. It might be, say, BCS 1203 or something. I just made that up. Then it's usually they do animal studies, mice, rodent studies, and so forth. And then when they determine that there may actually be some legitimate mechanism of action that would help human beings, there are usually phase 1 studies that are done in general on healthy young males, usually 18 to 25 or so, and they just want to make sure that the approach is safe. So one of the things we can say, first of all, right out of the gate, is that there's that harvesting bone marrow from the pelvic bone and introducing it into the spinal fluid through the low back area called the lumbar cistern is well tolerated and safe. And this has been published over and over again. So of course there are always sterile precautions that need to be taken. Just like any time you'd have surgery, you wouldn't want the surgeon to run to the bathroom and not wash their hands, But in general, the introduction of stem cells into the spinal fluid is well tolerated and safe. And again, this has been published over and over again. So then from that phase 1 trial, they go to phase 2. Phase 2 is sort of an exploratory trial in which they're looking at whether or not, beyond safety, is there a signal that this treatment is effective for the condition that's being studied. So is it effective for ALS? And usually they have what they call primary endpoints and secondary endpoints. So their primary endpoint is, in this case, say, there's a scale called the ALS Functional Rating Scale Revised, and we know I think it's a total of 48 or 50 points. Mm-hmm. But at any rate, unfortunately with ALS, we know, for example, that there's about a 20% 5-year survival with ALS. So if you think about, how many months in 5 years, you see that roughly speaking, there can be a decrease of about a point a month in the ALS functional rating scale. So as a person deteriorates, unfortunately, that the score on that rating scale goes down. Mm-hmm. So what happens then is when they do these clinical trials, they're actually looking at whether or not there's a change in the expected rate of decline in that scale. Right. So if you had somebody who wasn't on the treatment and they were getting worse by a point a month, so over, say, 6 months, they went down 6 points, whereas the person who got the treatment only went down 2 points or 3 points, that would be a significant improvement. Mm-hmm. So they had the phase 2 trial for ALS that was completed. And what they found in general was that there was about a 30% improvement in the expected rate of decline of people who received the active treatment, the neurone treatment, which was administered into the spinal fluid as well as into the muscles in people with ALS. And the protocol was basically 2 treatments over 6 months and then 6 months of post-treatment observation for monitoring for improvement or any adverse effects and things like that. Again, no serious adverse events. Now, there were secondary measures that basically said, are the stem cells doing what we expect them to be doing more at the biochemical or cellular level? So they measure what are called biomarkers in the spinal fluid, and they can determine if growth factors are in fact being secreted, if there's signal that what's being introduced in the spinal fluid is actively working on nerve cells in the brain. And in fact it was. So that was very positive. So there was a lot of optimism with the phase 3 trial, and the results were reported yesterday. So it was very, very very important for the 16,000 people currently suffering from ALS in this country. There's a big demand for this treatment. There is a lot of sort of the demand for what we call right to try, meaning people who just wanted the treatment regardless of whether it was proven to work, because it's such a terrible condition that, why not try it if it's safe? If it doesn't work, it doesn't work, but it's not going to really hurt anyone.
And there's not a lot of options.
Not a lot of options, really. Very few options, really, Zola and edaravone right now. So what ended up happening was very interesting, slightly disappointing, but still kind of optimistic. First of all, it failed. The study failed to show that there was a difference in the 2 treatment groups, meaning the active treatment group with neurone and the placebo group. Now that sounds kind of ominous, but if we take a little deeper dive and look at why and what actually happened, then you'll see, well, maybe it's not exactly as it sounds.
Right.
Because what ended up happening, first of all, is that the neurone treatment group had about a 32 or 35% improvement in the expected, 34.7 actually, in the expected rate of decline in the ALS rating scale. And so that actually reproduced the same results that we've seen with the phase 2 trial, and it's actually very similar to a South Korean stem cell product that's approved in that country by their FDA. But basically what we're seeing is that these stem cell treatments can improve the expected rate of decline by about 30 to 35%. So very positive, and one of the principles of scientific research is reproducibility, and I think we've seen reproducibility over and over again. And we've also seen safety. So once again, in the phase 3 trial, this treatment was well tolerated and safe.
Right.
Okay. What ended up happening was that the study, when they design a study, and they want to demonstrate a certain type of outcome, the statisticians who help design the study say, how many people do we have to have enrolled in this study to be able to show that there's a treatment effect? And part of that calculation includes what they expect to see from what is called the placebo group. So a placebo group is exactly what it might sound like. They are getting a treatment, but it's not actually the drug that's being investigated, or in this case, the stem cells. So these are people who volunteered for the study, knowing that they would have bone marrow extracted, but that when it came time to actually administering something into the spinal fluid, that maybe they just administered saline or salt water solution, that they couldn't see what was going on. And the studies generally are designed to be double-blinded, meaning the doctor who's doing the treatment also doesn't know, or they're separated from those who are evaluating the subjects in the trial afterwards to see if they improve. So if you know, for example, the person got the treatment, the active treatment, say neurone, and you were also the one evaluating that person using the scale, the ALS functional rating scale, you might be biased to sort of look for a positive response. So they really work hard to separate that out. There's almost always a placebo response. This is a fact of clinical research. And interestingly, this has become kind of a problem in clinical research because for a variety of reasons that haven't all been worked out, the placebo response in clinical research has actually been going up.
I agree with that.
Yeah. We've seen this in migraine trials where the placebo response can be as high as 40% of those treated. 40%. So it makes it very, very hard for pharmaceutical companies to show that this treatment that they're investigating actually works because the placebo response goes so high. So in the neurone trial, they powered the study for a 15% placebo response. So guess what happened? The placebo response was 27%. And it blew the statistics out of the water, making it look like the neurone treatment was not as effective, or was almost the same as the placebo group. Even though there was about an 8% difference, they have something called statistical significance, p-values, which basically say, is this difference enough to say it's not due to chance alone? And unfortunately, it didn't meet that statistical significance, even though the slowing and the decline of the functional rating scale was felt to be very clinically meaningful. And even though the neurone treatment performed very similarly to the phase 2 trial, very similarly to the South Korean trial, and they were able to look at those biomarkers in the spinal fluid and see that the stem cells were actually doing what they were expected to do. So this is very interesting, and it was hugely disappointing because it means that if nothing else, that the path to FDA approval in this country is certainly going to be slowed down. We don't necessarily think that Brainstorm Cell Therapeutics is going to cash in their chips, so to speak, and give up on their technology. We know that they plan to move forward, and we believe that they plan to continue to move forward with other potential indications for their technology. But it was a big disappointment for them. I'm sure a big disappointment for the principal investigators and the other people involved in the study, and certainly for the many people suffering from ALS who want this treatment. So anyway, I asked Michaela and Shauna to come on and talk a little bit about what it's like with our own trials and kind of what happens when people volunteer for trials and they understand that they may, in some cases, be getting a placebo treatment. It's not always the case for trials. Sometimes they have what is it called? An active open label, so that could be one. And then what's called an active comparator.
Oh,
Yep.
yeah.
Yep. And what is an active comparator?
Well, it functions as a drug that has a similar mechanism. Is that what you're talking about? Like the similar mechanism, they work the same way so that they can compare one it's on the market already, it's successful, and they compare their drug with that one.
Sort of a similar mechanism, or at least what ends up happening when we do clinical trials is there's something called good clinical practice. Mm-hmm. And that means that as a human being, we have to be very careful when someone is experimenting on you, right? There have to be good ethics involved. And so everything is very, very scrutinized. And we have what are called institutional review boards that review protocols for studies. But what happens with conditions like multiple sclerosis, what happens with conditions like epilepsy, for example, is when you have treatments out there that are proven effective treatments, and by effective, that doesn't mean cure, right, but it means, we know with some confidence that we can at least put a dent in the condition. So fewer seizures, slowing the rate of improved quality of life, slow relapse rate, progression, changes on MRI, et cetera, with MS. That at this point in time is considered largely unethical to have a placebo group when treatments are available. Mm-hmm. So the pharmaceutical companies who are investigating their product choose something to compare their experimental product with that is an approved product, because we can't have a placebo group when there is an approved treatment. Right. So we've done MS trials, and they use something called teriflunomide. Commonly, it goes under the trade name Aubagio, and so people who agree to be in these trials, they are blinded. They may get what is essentially Aubagio. They may get the study compound. So we just did several trials, and we're still involved with trials with something called ofatumumab or Kesimpta. And in the Kesimpta studies, they used Aubagio as a comparator. We're getting ready to do some trials with Sanofi Genzyme, and they will also be using the same teriflunomide in their trials. So it's kind of like they view that compound as one that they can beat up on a little bit because they're pretty sure that they can do better than that. But
But at least the patient would be on a drug.
They're on something, and that is also the case when they do epilepsy trials. They most of the new drugs coming out for epilepsy, we've had quite a few over the years, are now actually studied as add-on therapies because you wouldn't take somebody who has uncontrolled seizures and take them off their drug and risk them going into something called status epilepticus, which just means a seizure that won't stop. But this has a very high mortality rate. I mean, people die from status epilepticus. So you have to be obviously very, very, very cautious when it comes to this. So anyway, rounding things out, exciting, slightly disappointing news. You guys think?
Yeah. I think so. I think FDA is a very good thing, obviously. It's got very stringent rules. It wants to make sure that the patients are safe. But I think that because the rules are so stringent, sometimes you do have trials like this where it looks like it's not that positive, but really what they have to do is just change the endpoint, change their goal and what they're proving. It's still effective. But it just depends on the goals that they choose, what they want to prove.
Yeah. Now, the other thing about this, I want to put it out there, it's a slightly political statement, but not so much, is that when the Brainstorm Cell Therapeutics Neurone trial is going on, all of the sites for this study were major research institutions like the Mayo Clinic, Massachusetts General Hospital, and that's fine. These are highly respected institutions. There's nothing wrong with that. However, we have, somewhere in the range of 16,000 people suffering from ALS in this country. And they have the potential for an approved treatment that's a pretty high-tech treatment, harvesting someone's bone marrow, separating out the cells, growing those cells, manipulating them, that's the term we use, to express more of their neural potential, nerve cell potential. This has to happen at not just at these Massachusetts General, Harvard, medical school type places or Mayo Clinic. It has to happen at your community neurology clinic, right? We have to have these technologies available all around the country. There's just going to be a huge demand. As I said in the very beginning, $47 billion industry in the next 7 years. Huge demand. So I want you to think about, if you ever get in these conversations, say, it's wonderful that the Mayo Clinic or Harvard can lead the way. It's wonderful that Duke University can lead the way. But in the long run, what we want to hear is, can you go to your local neurologist and have stem cell treatments? And the answer resoundingly is, you should be able to, and yes. So we want to continue this conversation because we'll have some pretty exciting news along those lines for you at Charlin Health and Neurology. So thank you so much for tuning in today. I've taken more of your time than I intended, but we really appreciate you being on board.
Bye-bye.
Have a wonderful, wonderful Wednesday.